Hello? Anybody in there? Hello?
I'd like to invite you to the pajama party. Join me. It's late. Push pause on the video. Jump in your pajamas. Let's get comfortable and do a little nervous system. This is part 2, chapter 10, Hole's anatomy. So a little bit more detail on neurons. Here we have when the neuron cell body itself, so remember there's a cell body sticking off, there are dendrites, and there's an axon coming off. That's kind of the generic neuron. Well, the cell body portion, or the soma, when it's found in the central nervous system, we call this region the gray matter. You probably have seen pictures of the brain, or you will, I guess, and you'll see parts of the brain where part of it is gray and part of it is white. The gray part is cell bodies and unmyelinated axons, whereas the white is actually myelinated axons, the white matter. There's also something that we call nuclei, which are still cell bodies, but these are cell bodies instead of in the gray matter of the brain or spinal cord. We find these cell bodies within the white matter of the central nervous system. So it's dots of gray found amongst the white. And then we have ganglia, which are, once again, cell bodies, but these are now outside the central nervous system. They usually are just on the outskirts of the spinal cord. And one example would be the basal ganglia, for example. Ganglia are cell bodies outside the CNS. All right, so let's classify them. We've already done this in the previous one, but sensory afferent neurons go towards the central nervous system to deal with senses. And then this also accommodates proprioreceptors, which help to detect stretch, such as those in your leg for your knee-jerk reflex. Then your brain will respond through motor or efferent neurons to carry that message from the CNS to the effectors, glands or muscles. And then the last one that we have to talk about are interneurons. These are only found in the central nervous system, and they connect sensory and motor neurons. In the central nervous system, there are no sensory neurons, there are no motor neurons. You only find interneurons. Sensory and motor are found in the PNS. All right, now we have a diagram showing you. Here we have the sensory neuron, okay, going. Here you have a ganglion. You see the cell body of a sensory neuron outside the central nervous system, so we call it a ganglion. And it goes into the central nervous system. Here, this butterfly shape is the gray matter of the spinal cord, and the white outer region is the white matter. This is myelinated regions, and this is unmyelinated, such as cell bodies and unmyelinated fibers. Here in green, you see an interneuron that connects the sensory neuron to the motor neuron. Let me do one thing here real quick. I have to start a timer to make sure we have time. Okay. And then you see the motor neuron. Of course, this is the efferent cell in the PNS. All right. Now, there's different types of neurons. We typically use this as the generic neuron, but they don't all look like this, okay? Dendrites, cell body, axon, you see in blue here, the myelin. That's a multipolar neuron. There are bipolar neurons, which are much more simplified. Just a cell body, one axon, one dendrite sticking off. Dendrite sends the message, or receives the message to the cell body, and then passes through the axon, instead of many different dendrites coming off the cell body. And then lastly, we have unipolar neurons, which are even less common overall throughout the body, but they are common with the senses, where you have dendrites on one side that connect directly to an axon, and it sends it straight down the axon, as opposed to going through a cell body and so on. That's a unipolar neuron. Now we get fairly complicated. Don't get irritable. This is complicated, and you might get frustrated, but stay with it, work with it, and you'll be able to respond properly when I ask you a question about this on the test. And then that's irritability, ability to respond to a stimulus, conductivity to transmit an impulse. We did that in the previous chapter as well, in muscles. Now, what we're looking at is the neuron, focusing in on the plasma membrane of the axon. Here you see the axon, the inside and then the outside, and the pink part shown here is the plasma membrane. Now, the majority of the time your neurons are at rest. They're not doing anything, and they are prepared to send a message. At rest, we say that the neuron is polarized. Something that is polarized has an imbalance of charge. So you have north and south pole, or the differences in poles on a magnet. Polarized means differences in charge. And what you see in a neuron is that at rest, the outside is positively charged and the inside is negatively charged. And we'll talk in a second about why. Actually, not in a second, right now. Inside the axon, there is fewer positive charges, and there's lots of negative charges. Those negative charges are mainly contributed from DNA and proteins. DNA is very negative. You don't see it in this picture because it's in the cell body, and we're looking here at a cross-section of an axon. But it's very, very negative inside because of DNA and proteins, even with all these potassium ions inside. And then the outside is very positive. That's due to sodium. So again, at rest, the inside is negative. In fact, it's negative 70 millivolts. Don't pay attention to this number. It's generally negative 70 millivolts. All right, so now when a message wants to be sent down the axon, here you see zooming in on the axon, we have what's called depolarization. And when depolarization happens, it's in response to a stimulus, and some channels in the membrane of the axon open, specifically that fit sodium. And sodium enters the cell, and we call this depolarization. And sodium brings with it positive charges, and that makes the inside of the cell in this one area positively charged. Okay? As this positively charged ion enters, it triggers neighboring channels to also open, which allows sodium to enter, and you can see that here. So this whole region of the neuron is depolarized, and it's going to trigger this region of the neuron to become depolarized, which will trigger this region of the neuron to become depolarized. And again, by depolarized, that just means sodium entering the cell. And this passage of positive ions going down, 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 down the axon is called an action potential or an impulse. Here's another picture showing sodium entering the cell through sodium channels. The outside here is positive, and now that positive is brought to the inside, and you can see where the sodium enters is positive, where the other regions are negative, still negative 70 millivolts, where this rises into the positive. And we'll talk later about what that means. So we have an action potential, okay? Once it starts, it's going to continue all the way down the length of the axon, from all the way to the axon buttons or axon terminals. We call this all or none. When an action potential starts, it happens. You cannot stop it. You cannot block it. It automatically goes all the way to completion. And then as the action potential is traveling down the axon, we start to reset the cell and prepare it for another impulse, another action potential to be sent immediately after, should we want it. So after depolarization passes, we start repolarization. And repolarization happens when the potassium ions that are inside leave the inner part of the cell and go to the outside, to the extracellular fluid. And again, we call this repolarization. We're taking the cell back to its polarized state to repolarize it. And then after repolarization, you'll see we'll talk about there's a special pump called the sodium-potassium pump that uses ATP, and it helps to restore the proper configuration or the proper arrangement of sodium on the outside and potassium on the inside at rest. Because after an action potential goes, after depolarization and repolarization, the ions are in the wrong spot. Let's show you what I mean by that. Here we are at rest, negative inside, positive outside. There's lots of sodium out here, lots of potassium in here. Stimulus occurs, depolarization, sodium enters. Makes the inside positive, that triggers neighboring channels to open, more sodium enters, more positive. Now behind, we start to repolarize. The sodium channels have closed, and the potassium channels have now opened and allowed potassium, K+, to leave. And that's going to repolarize the cell. Here you see in yellow, that's yellow. That would be green. The green area of the cell is repolarizing, and then the yellow area is back at rest. And to go from this green that you see here to the yellow, this is where the sodium-potassium pump works, because you'll notice that we have lots of potassium outside, and the sodium was inside. We want to take this potassium back in, and we want to take the sodium that's in here back out. And this is where the sodium-potassium pump works. It takes sodium out in exchange for potassium to come back in. And you see that working here. Okay, this is a complicated diagram that I really like, showing a axon at rest. Here's the inside of the axon. Here's the outside, the extracellular fluid. You see a measurement device, a needle stuck into the cell, and this is perfectly possible, measuring the voltage inside, and it's negative 70 at rest. Here you see the inside is negative because of proteins that are negative and DNA. So it's negatively charged inside. That's what those dashes mean there. And what you're going to notice here is the sodium-potassium pump is constantly working to maintain that negative 70 millivolts. So it's going to take 3 sodiums out of the cell in exchange for 2 potassiums in. And this is going to build up a big, big gradient of potassium inside, and it's going to build up a big, big gradient of sodium outside. And the important part of this is it's like creating a dam and building up a bunch of water over here in that dam. so that when you open the dam, the water rushes down, and you use that as the signal or the force in a way. But then if you want to use that signal again, you want to use that force again, you have to move all the water back to the other side of the dam to build up that gradient or that very large amount of water. All right. Now, there are some neurons that are myelinated and there's some that are unmyelinated. Myelin is, in the peripheral nervous system, done by Schwann cells. In the central nervous system, it's from oligodendrocytes. That's some review for you from the previous section. But the purpose of the myelin that you see here shown in red is that it makes the impulse, the action potential, travel faster. And that's because the only channels that are working are in the nodes of Ranvier, the regions of the axon where the channels don't function. The region of the axon where the channels do function, because where the myelin is, it blocks those channels and they don't work. So sodium can only enter here, and when sodium enters here, it depolarizes this region of the membrane. But because none of these channels in here work, this is going to trigger the channels over here to open, and sodium's going to enter. So in a way, the signal is jumping, or we call it saltatory conduction. Saltar in Spanish means to jump. And the signal jumps from node to node to node, as opposed to going open, 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 open. Instead it goes open, 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 and it's much, much faster. So myelin makes the signal travel faster. Here again is showing you the same thing. Here shown in yellow is the myelin. Here you see the nodes of Ranvier, and sodium has entered, making the inside positive. And here I have a question for you. Going back a step, a stimulus arrives, the cell depolarizes, and that depolarization passes through the cell. After depolarization, immediately in each section of the neuron, you have repolarization, where the potassium is moving out. And then right after repolarization, there's a special case or a special period called the refractory period. And during the refractory period, you have the sodium-potassium pump working to reset gradients. But also during that period, you cannot send another action potential. So no matter how hard, no matter how fast or strong the stimulus is, another action potential will not be sent in that neuron for that brief split millisecond. So I want you to think about what the purpose of the refractory period is. Okay, this is a difficult question, but I really want you to think about it. Don't just pass on. Think what the purpose is, and of course some of you may search it, which is okay, but I'm going to ask you this question when you come into class, of course. All right. So then when we get to the end of the axon, we get to a synapse, and the synapse can be onto another nerve or onto a muscle, as you know. And to get across the synapse, we have a neurotransmitter, such as acetylcholine we saw in the neuromuscular junction, that has to pass across. And the acetylcholine will bind to a receptor on the postsynaptic cell, or the next neuron in line, and stimulate that neuron to send a message, to send an action potential. So here's a diagram showing that one axon synapsing with another one. Here it shows blown up. These little purple things shown here are the receptors. Here's the vesicle with neurotransmitter that connects to the plasma membrane and releases the neurotransmitter out. These will then bind to these channels, and these are sodium channels typically, and it will allow sodium to enter into the cell, and it'll depolarize this postsynaptic cell that currently is at rest, and it's the receiving neuron. Then the neurotransmitter will get broken down, chopped up, won't work anymore, and then the postsynaptic cell won't be stimulated anymore. It'll be done. So now what I'd like you to do, I'm not going to click on it, but I'd like you to go to the slides, or you can just type this in somehow, and I'd like you to watch this animation. It goes through all the stuff that we've talked about, and it's actually a really good animation. You'd spend a good amount, good 3, 4, 5 minutes doing that, and I think it'll really clarify your understanding. All right, you stay classy, San Diego. I'm Ron Burgundy. I'm Ariel Haas. Wait, damn it! Who typed a question mark into the teleprompter?